Hello everyone, welcome back to my channel. Today's topic is biosensor detection of pollutants. Definition is biosensor is an analytical device consisting of immobilized layer of the biological material, that is enzyme, hormone, antibodies, organelles, nucleic acids, or the whole cell in the intimate contact with a transducer, that is a sensor, which is a physical component which analyze the biological signals, that is it detects the presence of a biological material and convert it into an electrical signal. So here a sensor can be anything, that is a single electrode and a photocell or a thermistor. So what we can say here is biosensor construction is a three-step process and it involves combining the two elements with different characteristics. Here first a biological sensing element is chosen, then a transducer is selected, and finally the biological component, that is the detection element, okay, is fixed to the transducer, and then it will be converted into an electrical signal. So microorganisms have a huge potential for the detection of a wide spectrum of chemicals, okay, and the substances and their mixtures. They are adjustable to different reaction conditions, and compared to the enzymes or the antibodies, they do not require, that is microorganisms do not require expensive preparation process. They can be genetically modified too, hence they are, we can say, useful for the detection of the pollutants. Then the principle of the biosensor, basically, so a principle of biosensor is quite simple. The biological material is immobilized as an immobilization support. The permeable membrane in the vicinity of the sensor, that is it is applied there. The substance to be measured pass through the membrane and interact with the immobilized material and yield the products. So, a product may be heat, gas, oxygen, electrons, hydrogen ions, or the product of ammonium ion, that is NH3. So, the product passes through the other membrane to the transducer. Then, the transducer convert the product into an electrical signal which is amplified. So, the signal processing instrument convert the amplified signal into a display, most commonly the electrical signal, which can be read out and recorded. So this is the basic, we can say, diagram or the schematic outline of a biosensor, which here shows the basic principle that is utilized in case of a biosensor. So here it is a substrate, it is the immobilized membrane where enzyme, antibody, or any cell is used, okay, only one of those is used at a single time. So it will comprise of the biological component here, again the membrane, the, we can say, out of these any one is loaded here, then the product is formed, and then here transducer is used, that is a physical component, then amplifier is used which will give signal here, okay, that is microelectronics, that is a device is installed here, okay, that will process the data, then digital readout is given, and at last recorder will give the signals or the recorded material. So this is the physical component in case of a biosensor. So now types of biosensors, that is biosensors are of different types based on the use of different biological material and the sensor devices. So a few of them are discussed below. First is the electrochemical biosensor, then amperometric biosensor, then thermistor containing biosensor, bioaffinity biosensor, optoelectronic biosensor, and a whole cell biosensor. So whole cell biosensor is also known as microbial biosensor. So first is electrochemical biosensor. So this type of biosensor have been developed by using electrical devices such as the field effect transmitter or LED, that is light emitting diode. So basically it is made up of these type of materials. Then amperometric biosensors. So amperometric are those which measure the reaction of an analyte with enzyme and generate the electrons directly or through a mediator. So basically in case of amperometric biosensor, electrons are generated. Amperometric biosensor contain either enzyme electrode or without a mediator, or chemically modified electrode is used here. That is, two conditions are there. So the oxygen and the peroxide based biosensors are the example of amperometric biosensor. So this is a diagram of a glucose electrode. So here glucose solution is used. Okay, then this is the saturated KCl solution which is we added here. Platinum oxide is used. Okay, the cellulose acetate membrane is present. Immobilized glucose oxidase, that is an enzyme that is used as a biological component, is the immobilized glucose peroxidase. And the permeable Teflon membrane is also present, and a ring seal is also there. So again, the next diagram is a mediated biosensor, or we can say the type of a amperometric biosensor. So here electrode is used in by which electrons are released. It interact with the enzyme, that is the biological component here. Okay, it interact with the redox mediator. Okay, electrons are attracted towards electrode. Okay, and the product is formed by the conversion from the substrate. So this is a mediated biosensor. So now again is the thermistor containing biosensor. So thermistor is used to record even a small temperature changes, that is between 0.1 to 0.001 degrees Celsius during the biochemical reaction. So thermistor is basically a device to analyze the variations in the temperature. So by immobilizing enzymes, that is like cholesterol, okay, oxidase, glucose oxidase, invertase, tyrosinase, etc., thermistor have been developed. So basically these enzymes are used in thermistor. Moreover, thermistor are also employed for the study of antigen antibody with very high sensitivity in case of thermostatic enzyme linked immunosorbent assay, that is in ELISA test we can say. For so in ELISA also thermistors are used for detecting the interactions between the antigen and antibody. So this is a diagram, basically simple biosensor combining with electrode, chemical electrode, and an enzyme that is immobilized to a semi permeable membrane. So here platinum anode is used. It is the electrode that is used. Cathode is here. Then this is the substrate or the analyte which should be detected, that is in any natural conditions. Enzyme is put here as a immobilized material. This is the semi permeable membrane. Okay, so this is it. Now the next is bioaffinity biosensors. So bioaffinity sensors are developed recently. Okay, it measures the concentration of the determinants, that is the substrate, based on the equilibrium binding process or the mechanism we can say. This show a high degree of selectivity. Okay, that is it selects only specific we can say determinants. These are of diverse measure because of the use of radio labeled, enzyme labeled, or the fluorescence labeled substances that are used here. So they possess specificity. Okay, principle of bioaffinity biosensor is shown here. So this is the diagram. Determinant analog, labeled receptor. This is the determinant that will attract the receptor. This is the transducer. Then in this biosensor, a receptor radio labeled and allowed to bind with the determinants analog, immobilized onto the labeled receptor, form an intimately bound. That is here we can say radio labeled receptor is basically used that is intimately bound with the complex with the determinants. So this type of biosensor are the use of lectin receptor for saccharide estimation, hormone receptors for hormone, drug receptors for a drug, antibodies receptors for antigens, and the nucleic acid that is act as a gene probe for the inherited disease and the fingerprinting. So basically these are, we can say, receptor specific in nature, that is designed for a particular substrate. Then optoelectronic biosensors. So in these biosensors, either enzymes or the antibodies are immobilized on the surface of a membrane for measuring the color. Biosensor with enzyme and dye is immobilized to a membrane. So here both biosensor is used, okay, that is with enzyme along with a dye. Then whole cell biosensor, it is also known as microbial biosensor. So in this device, either immobilized whole cell of microorganisms or their organelles are used, okay, full or whether full cell or the part of their cell, that is organelles. So these react with large number of substances and show generally slow response as compared to others. So example of some microbial biosensors, that is where microbes are utilized, is Brevibacterium lactofermentum sense for assimilate sugar, that is for detection of assimilate sugar, Methylomonas flagellata sense for the methane, and Trichosporum brassicae sensing for the acetate and ethanol. Now, the applications of the biosensors, that is biosensors have wide variety of applications, that is it is used in various fields. So first is used in medicine and health. So biosensor have tremendous potential for its application in the field of medical science, that is a device, we can say mini pump is filled with insulin, has been constructed to deliver insulin to the diabetic based on the glucose levels of the blood. It is used to treat insulin, okay, we can say deficiency in the diabetic patients. So it is also used to detect mutagenicity of the cancer causing chemicals also. Then used in the pollution control. So biosensors are also very helpful in the environmental monitoring and the pollution control, that is it is useful in detection of water pollutants, soil pollutants, heavy metals. So as far as the quality control of the drinking water is concerned, the monitoring of the biosensors are successful in monitoring of pesticides in the water. In Japan, a biosensors coupled with oxygen electrode and immobilized trichosporan cutaneum is used for measuring BOD, that is biological oxygen demand. Then uses in industry. So generally spectro photometer and autoanalyzers are used to estimate the substrate utilized and the product formalin fermented broth. So the biosensors can be designed to measure in the fermentation product. Okay, another biosensor has designed in Tokyo for measuring the odor, the freshness and the taste of the food. Then biosensors in the military. So the darker side of the biosensor application is to provide support to the military with such a biosensor that can detect toxic gases including chemical warfare and the agents. Such biosensors have advantages over the traditional methods of the sensing of the chemicals, and that's why they are utilized in these fields. So yeah, this is all about biosensor detection of pollutants. Hope you will like my video, and if you like my video, please do like, share and subscribe to my channel.